Hello everyone, this is Dr. Nidhi from Biotechnica. I welcome you to this session. In the following session, we are going to have a discussion over the questions those have been asked from Unit 3 in the recent CSIR exam paper held on 17 September 2022 in Shift 1. If you have watched my video under how to what to series shared by Biotechnica about Unit 3, so there I had given you this idea that nearly 10 to 12 questions they are asked from Unit 3 in CSIR exam paper. So as expected, 11 questions they were asked, and as expected again, some of those questions were combined with the concepts from other units like Unit 2, Unit 4 or Unit 8. Okay, so let us continue and let us find out that what was the level of the questions and what the answers given by us, they match up to the CSIR answer key. So here is our question first, right, with question ID 8. And this question is about tRNA. Basically, this is from translation, that which one of the following statements about the recognition of tRNA by their cognate aminoacyl tRNA synthetases is correct, right? So whenever I teach translation, I always tell my students that besides tRNA, mRNA or ribosomes, aminoacyl synthetases, the role of aminoacyl synthetases, that is equally important, because these enzymes, they are responsible for adding up the right amino acid to the tRNA. Okay, so now this question asks that which one of the following statements about the recognition of the tRNA by their cognate, means related aminoacyl tRNA synthetases is correct, right? Now here the option 2 is the right answer. Okay, option 2 says aminoacyl tRNA synthetases, they recognize their cognate tRNA by recognition of their anticodons in some tRNA, right? Now what do we mean by that? For that, let me show you this slide with the explanation and the diagram of tRNA. So if you know about the clover leaf-like structure of tRNA, so tRNA has an amino acid arm, then a T arm, then anticodon arm, and dihydrouridine arm, D arm. Okay, so aminoacyl tRNA synthetases, they attach the correct amino acid to their tRNA. Right. Now, an individual aminoacyl tRNA synthetase must be specific not only for a single amino acid but for certain tRNAs as well. Now, you might think that how aminoacyl synthetase will be able to attach the right amino acid to the tRNA, and most of the students, they know that, okay, anticodon loop has a role in it, right? However, let me tell you that except the stem, this one, amino acid arm, this is very important, right? This is going to determine that which amino acid will be attached to the tRNA. So it's not like that all the nucleotides which are present in the anticodon arm only, they are responsible for the attachment of the right amino acid to the tRNA, right? Because some nucleotides, they are conserved in all tRNA and therefore they cannot be used for discrimination. Nucleotide position necessary for discrimination by aminoacyl tRNA synthetases seem to be concentrated in the amino acid arm and the anticodon arm. Okay, including the nucleotides of the anticodon itself, but they are also located in the other part of the tRNA molecule. Like I give you one example. Let us take example of serine. So 6 codons, they are available for serine. Then 6 anticodons would be there. That means 6 tRNAs would be there. Those all will be iso-accepting tRNA. So how one aminoacyl synthetase will be able to recognize all the tRNA which are having different anticodons. Okay, so I hope you got the idea that why here option 2 should be the right answer. Okay, now let us move to our next question. Question ID 10. Now this question asks that which one of the following statements made about the bacterial replisome is incorrect? Yes, bacterial replisome. So this question is definitely from replication. And here, if we read out the statements 1, 2, 3, 4, okay, so you will find that option 4, statement 4, that is the right answer. That is incorrect, means that should be the right answer here for this question, because this statement is incorrect. This statement says that E. coli OREC carries repeats of 2 sequence motifs, repeats of a 9-mer that collectively form the site, at which the origin first becomes single-stranded and repeats of 13 mer to which the DNA initiator protein binds. Right, now why this statement is incorrect and how you could have been able to solve this question? Definitely, if you know about the replication initiation process for prokaryotes. Okay, now to make you recall, I am showing you this diagram here. Right, so this is about the ODC sequence of bacteria. Okay, so ODC, that is a conserved region, right, where the replication would begin. Okay, now this region contains 4 9 base pair binding sites for the E. coli initiator protein DNA. So DNA is the initiator protein, right, which will be helping with the initiation of replication in bacteria. And here 3 repeats of 13 base pair sequence you will find. Okay, so the one which have been shown in blue color, these are 13 mer repeats, and similarly you can see 4 9 mer repeats they are present. So that what has been mentioned in the statement 4. However, there they have mentioned that repeats of a 9 mer that collectively form the site at which the origin becomes single stranded. Now that is incorrect, right? Why? Because the initiator proteins, which I have just explained, they will be binding to the 9 mer site, and the 13 mer site, that will become single stranded, right? So yes, half part of that statement we can consider that is correct, but the other half is incorrect, right? So DNA is becoming single stranded in the 13 mer region and not in the 9 mer region. So that makes statement 4 as an incorrect statement. Okay, so I hope you were able to solve this question. This is easy. Okay, now let us move to our next question with question ID 11, right? Here, the question asks that which of the option correctly matches the proteins involved in transcription? Column A with the DNA binding domains they carry, which have been given in column B. Right, now you could see here that some factors they have been mentioned, and here we have the DNA binding domains. So to answer this question, you should have a fair idea about that what actually DNA binding domains are. Okay, so I give you a gist. Okay, here answer would be option 4, but to know that why it should be option 4, I'm taking you to the next slide. And the proteins which are involved in the control of transcription, the transcription factors, right? So basically they have 2 loops, right? 2 domains, right, which are known as DNA binding domains and transcriptional activation domain, right? Now, DNA binding domains, they help these factors to bind to the DNA. And the transcriptional activation domain, they help these factors to interact with the other components of the transcriptional machine. So we'll find that these factors, they have some structural motifs, right, in or as DNA binding domains. So those structural motifs, they have particular sequences of amino acid, right? So amino acid, they will be present in a specific sequence in a specific manner. So that is known as the DNA binding domain. Okay, the DNA binding domain, they could be of different types, like zinc finger motif, if you have heard of zinc finger motif. So here what happens, as you could see, that within the polypeptide, 2 cysteines and 2 histidine, right, in 2 different parts of the polypeptide, they'll interact with each other, right, through a zinc ion. So the stretch which is in between, right, the peptide, which is the stretch of the peptide which is in between these 2 pairs of amino acid, that will jut out like a loop. So this loop is known as zinc finger. Okay, this helps in binding to the DNA. Similarly, we could see helix turn, helix motif is there. Okay, so here there are 3 helices. 3 helices, those are formed, and they are further joined to each other by turns. So this is known as helix turn, helix motif, right? So the various studies, they have actually proved that the helix, which is towards the carboxyl terminal of the polypeptide, that helps in binding to the DNA. Okay. Next, we have helix loop, helix motif. So as you could see here, the stretch of 2 helices of the amino acid, which are separated by a loop. Okay. Now, this kind of motif that could help in dimerization to the polypeptide, right? Next, we have leucine zipper. So what do we have in leucine zipper? The stretch of the amino acid, which will be having leucine at every 7th position, forming a zipper. Again, here you could see that two polypeptides having leucine zipper, they could form dimer with each other. Okay, so helix loop, helix motif, leucine zipper motifs, they help the polypeptides to form homodimers or heterodimers. So I hope you got an idea that what all DNA binding domains are. Now next you had to recall that where these domains means what all factors they do have, what all kind of DNA binding domains. So that is something which you need to memorize. So I'll not say that this question is very simple. We can keep it as a moderate question because you had to recall that whether these factors, they are having ring finger or loop helix or the other type of DNA binding domain. So here ring finger domain are one of the most common structural motifs, right, in eukaryotic cell, which employ the motif in some of their most important proteins, including TF3A. Then MyoD, right? Now MyoD, actually this is involved in muscle development, right? Muscle development that is controlled by the MyoD family of the basic helix loop, helix DNA binding proteins, and these proteins dimerize with ubiquitous products of E2A gene, E12 and E47 and bind in a sequence specific manner to enhancer regions of muscle specific genes, activating their expression. So basically here MyoD, they have basic helix loop helix, okay, and basic leucine zipper domain of C June junction functions in transcriptional activation through interaction with the N terminus of human Tata binding protein associated factor, that is human TAF. And then Crow C1 type. So I am sure everyone remembers that Crow C lambda repressor, right? So this has HTH domain as the DNA binding domain. Okay, so that makes option 4 as our right answer. Okay, so now let us move to our next question and let's find out that what do we have there. Question ID 58. Following statements were made about chromatin remodeling in eukaryotes. Okay, so this is about chromatin remodeling, and this topic, this question has been picked up from the topic transcription. Okay, so chromatin remodeling completely alters and or slices the nucleosome but cannot displace it. So you have various statements which have been given, and we have been asked that which one of the following represents the combination of the correct statements. So answer is option 3, that means B, C, E. These 3 statements, these are the correct statements. So let's find out that why these statements are correct. So B, yes, what do we have in statement B? Chromatin remodeling is an energy-driven, developmentally regulated active process. So that is correct. What is chromatin remodeling? Chromatin remodeling is bringing some changes in the architecture of DNA so that the transcriptional machinery can reach the important sequences which are present in the gene to complete with the transcription. Especially if we talk about the eukaryotes, right? In eukaryotes, we know that DNA that is tightly packed with histones, right? And that is existing in a compact chromatin. So that chromatin has to be loosened so that the components of the transcription machinery can reach there. So yes, chromatin remodelers are there which can help with the chromatin remodeling, and chromatin remodeling, that is an energy driven process. So yes, statement B, that is the correct statement. Then we have histone acetylation is a reversible process in which each direction of the reaction is catalyzed by different enzyme, that has been given in statement C. So this is also right? Histone acetylation, that is very important. Whenever I teach, I always tell my students that don't miss on histone acetylation, HAT activity. Okay, so that helps with the transcription. Then next we have a phosphorylation of serine 1 of histone H2A has been associated with transcription repression. So this is again correct. So here phosphorylation of histone H2A on serine 1 by mitogen and stress-induced kinase MSK1 that is known to inhibit RNA synthesis. Okay, so therein, as we have already discussed, that chromatin remodeling, which is needed for the completion of the transcription in eukaryotes, right? So hat activity and all, this is important so that the transcription can happen easily, so that the DNA could get access, the transcription machinery could get access to the sequences within the gene. All right, so I hope you were able to solve this question. Okay, so this was also simple. It was not that difficult if you know about the hat activity. Next question, question ID 61. Okay, so this is again from replication, but if you will read this question, you will find that the concept of replication is merged with cell cycle. I always tell this, that replication can always be merged with the cell cycle concept to frame questions for part C. Okay, so what do we have in this question? In eukaryotes, DNA replication must occur with extreme accuracy and only once to prevent the damaging effects of gene amplification. The following statements were made regarding possible mechanism involved in achieving tight regulation of DNA replication. Right. So let me tell you that the initiation of replication in eukaryotes, right? Earlier we had seen one question that was based on the replication initiation in prokaryotes. Now this is the question which is related to the eukaryotic replication initiation process, and we have one statement, right? The statement A, which had been taken from cell cycle topic. So here what do we have? High APC activity in mitosis, an early G1 phase of the cell cycle that triggers the destruction of CDT1 inhibitor geminin. Yes, thus allowing CDT1 to be active in early G1 to load helicases, right? So let's find out that what is the right option here, so that further I can go further to explain that why we have A, B, and D statements as correct. So yes, these three statements are correct, A, B, and D. Now why statement C is incorrect, right? So let us make it easier and let us find out that why statement C is incorrect. MCM helicase loads at the S phase of the cell cycle, so that replication begins only at S phase. Yes, we all know that replication that begins at S phase. And MCM, these are the helicases, eukaryotic helicases, right? But the helicases, they are loaded at G1 phase. Okay, so they are not loaded at the S phase. So that makes this statement as the incorrect statement, right? So this part is again very important, right? How the loading of helicases that is done at the G1 phase, but their activation that occurs during the S phase, so that the replication can happen at the S phase, right? So helicase loading at G1 and helicase activation at S phase. Now let me tell you that helicase loading, that is done with the help of some factors. Those are in common. In general, they are known as licensing factors. Okay, so they are known as CDC6, CDT1. Okay, now CDC6, CDT1 proteins, they load the MCM helicase to the ORC, origin of recognition complex. That is a complex which helps with the initiation of eukaryotic replication. Okay, so statement C is incorrect. So we have already discussed that why statement C is incorrect, because loading that is being done during G1 phase. Now CDT1, this is inactivated by the protein geminin, right? Now, if CDT1 that will get inactivated, so how the helicase that will be loaded? Okay, so that means geminin that should not be there during G1 phase, right? So geminin, basically it is an APC-C target. Now what is APC? So those who have learned cell cycle, they might be having an idea about that. APC, anaphase promoting complex. So it targets many proteins, right, which help with the progression of the cell cycle. It means which are related to various molecular events related to the cell cycle. And its levels therefore increase in S and M phases when APC-C is inactive. So A is correct. Yes, so A is correct. So I'm going back to show you that what the statement A is all about. High APC-C activity in mitosis and early G1 phase triggers the destruction of CDT1. So again, I repeat with this, CDT1 that should be there so that the loading should happen. Okay, so CDT1 inhibitor geminin that shouldn't be there. So that what has been given in statement A. So that makes this statement is a correct statement as well. Okay, so this was an easy question, right? It was not that difficult if you have learned cell cycle, if you have learned eukaryotic replication in detail. So this was very easy to be answered. Okay, now let us move to our next question, and this question is about DNA repair systems. Yes. So DNA repair, that is another important topic, right? We have to cover from unit 3. And every time, every year, in every paper, we see that one or two questions, they could be picked from DNA repair topic. Okay. Now here this question asks that excision repair system replace a short stretch of DNA around the site of damage. The following statements, they are made about nucleotide excision repair. Yes. So this question is about nucleotide excision repair in E. coli, and you had to tell that which of the following options, right? Those are having different combination of the statements. So that is correct. So let's find out what was the right answer. So here, as per CSIR answer given is option 4, that is B, C, and D. But we can challenge this question, right? Why? Because here only statements C and D are correct. Okay. Now, why I'm saying so? Because just look at statement second, right? Statement B. So here it has been mentioned that 50 to 60 residue long stretch of DNA between the two nicks is removed by the action of UVRD. So if you have learned the nucleotide excision repair system, so you might be knowing that we have the proteins which help with commencing this nucleotide excision repair mechanism, and those are UVR A, B, C, and D. So UVR A-B complex that helps in the recognition of the lesion, and further UVR, UVR-A gets released, and UVR-B dimer that recruits UVR-C. UVR-C further it create two incisions, one located 4 or 5 nucleotides 3' to the lesion, and the other 8 nucleotides 5' to the lesion. So approximately 12 nucleotide long stretch that is removed. Okay, I repeat, approximately 12 nucleotide long stretch that is released, of course including the lesion. Okay, whereas here it has been written that 50 to 60 residue long stretch of DNA between two nicks is removed by the action of UVRD. So this should be incorrect. So our right option should be 3 only, because C and D they are correct. The gap which is generated, that is filled by DNA polymerase 1, and the distortion caused by the lesion, that is recognized by UVR-A-B complex that I have already mentioned. Okay. Now let us move to our next question, question ID 64. Here, this question is about the role of introns. Yes, the role of introns. Now, the question says, although introns are not a part of the process transcripts that gets translated, they are important for several reasons. The following statements are made with reference to the possible ways in which introns are crucial to the cell survival. So basically, you had to find out that what are the roles of the introns among the given ones, right? As in the statements A, B, C, D, E. So correct answer was option 3, A, B, D, E. Right. So what do we have in statement A? They permit the generation of different protein products from the same gene. Yes, if you know about the alternate splicing, so yes, they actually permit the generation of different protein products, because mRNA splicing would be done in different ways, and hence we may get alternate forms of the proteins from the same gene, right? Next, we have, they may encode miRNAs which modulate the expression of genes. So this is again correct, right? Why? Because introns, they harbor several kinds of non-coding functional RNA genes. miRNAs, siRNAs, PV interacting RNAs, long known coding RNAs, and even small nucleolar RNAs. About half of the miRNAs in the human genome are in introns. Okay, so that makes our statement B a correct statement, right? Now moving to statement C. They often encode peptides which which play a role in regulating gene expression. So this right. So this word is incorrect. So remember, an intron is a region that resides within a gene but does not remain in the final mature mRNA molecule following transcription of that gene, and it does not code for amino acid that make up the protein encoded by that gene. Thus, statement C is incorrect. Okay. Now let us find out that why the other two statements, statement D and E, they are correct. So if you know about the nonsense mediated decay mechanism, okay, only if you know about this mechanism, so you could have been able to find out that why the other two statements given will be correct. Right now, this mechanism basically it is a surveillance mechanism. Okay, so cell has to evolve many surveillance mechanism to check with those mRNAs, those are having errors. Okay, so those are targeting. So one of the very efficient, one of the best studied RNA surveillance mechanism is this one, nonsense mediated decay mechanism, which targets the mRNA those harbor the premature termination codon, that is PTC. Okay, so if an mRNA is having pre-termination code, so that will be targeted by the NMD, nonsense mediated decay mechanism. Okay, now this is signaled by the presence of the exon junction complex. Now, what is exon junction complex? This is a multi-subunit protein complex which is deposited 20 to 24 nucleotides upstream of an exon-exon junction during pre-mRNA splice. Right? So that what I am repeating, that this mechanism, this pathway, you should be knowing if if you want yourself to be able to solve the questions based on the EJC, that is exon junction complex, or nonsense mediated decay mechanism. Now, here in statement D, we have they promote export of certain mRNA through the recruitment of transport proteins by exon junction complex. So like I have explained, exon junction complex, that is a multi-subunit protein complex which is found at the junction of two exons, right? When splicing occurs, two exons they get ligated. So there this EJC that will be placed. Now this EJC, exon junction complex, that helps with the export of the mRNAs. Okay, not only that, through this nonsense mediated decay mechanism, it will be helping with targeting those mRNAs, meaning decaying those mRNAs, those are having PTC. So because of that reason, these two statements, they are correct. Okay, so this EJC that is placed, and if there is no PTC, if there is no pre-termination codon present, so this exon junction complex that will be removed. However, if there is PTC, that is a pre-termination codon present, so EJC that will be retained, and nonsense mediated decay mechanism that will work. So again, that was not a very easy question, I would say, right? So yes, we'll keep this question in the category of moderate questions. Now let us move to our next question, question ID 65. Here it has been mentioned that initiation of transcription is a complex process involving promoter recognition, conversion of the initiation complex from closed to open form, abortive initiation events, and finally promoter escape. The following statements are made regarding these steps in transcription initiation. So another question we do have from transcription, and this is related to promoter escape and the other molecular events which are related to the transcription initiation. So again, when I teach transcription during my classes, I always tell that promoter escape, promoter clearance, abortive initiation, that is a very important topic to be covered. So yes, let me know whether you were able to solve this question. Yes, yeah, that was an easy one, right? So what is the right answer here? Answer is option 1, A and C. Okay. So what do we have in statement A? Promoter escape in bacteria is usually accompanied by the release of the sigma factor from the RNA polymerase holoenzyme complex. Okay, so those who don't know about it, let me tell you that RNA polymerase in prokaryotes that has a sigma factor bound to the core RNA polymerase unit, transforming it into RNA polymerase holoenzyme. Okay, so that the sigma factor helps with the recognition of the promoter and it can further carry out the transcription. However, the sigma factor gets released, right? When RNA polymerase leaves the promoter, that is promoter escape happens, so this sigma factor gets released. So that's what we have been mentioned in statement A. Okay. And then why statement C is correct? Because promoter escape in eukaryotes that is accompanied by the phosphorylation of RNA polymerase last subunits on is CTT. Right now, this is about eukaryotes. Okay, in eukaryotes again, this part is important about how the initiation of transcription that is related to the phosphorylation of one of the subunits of RNA polymerase 2 at CTT domain. Okay, so yes, this statement is a correct one. Right. Now, few students had asked me that, ma'am, why statement D that is not correct? Okay. So here what has been mentioned, let's read out this statement to avoid any confusion. Promoter recognition in bacteria that is governed by the sigma factor which binds to the -10 and -35 regions of the promoter, followed by recruitment of RNA pol to core enzyme. Okay. So here comes the incorrect word. Right. Here what we are finding RNA pol 2. And here what do we have? -10 and -35 regions. So they have fused the facts from prokaryotes and eukaryotes together. Okay. So that makes this statement as an incorrect statement. So that was an easy one, right? Now let us move to our next question. Question ID 66. Fidelity of protein synthesis. Okay, so this is again related to translation. See how beautifully they have covered all the fundamental processes. There we have questions from replication, we have questions from transcription, we have questions from translation, then cell cycle concept that has been merged. Further, I'll show one question where mutations they have been added. Then we have questions from DNA repair. So the entire unit that has been covered. So this thing also I mentioned that we cannot miss any subunit from unit 3. So whenever you prepare unit 3, so remember you have to cover all the subunits because the question they could be created in this way where the concept they could be merged from all the subunits together. Okay, so coming back to our question. Now this question is about that how amino acyl synthetase that adds up the right amino acid, right? We have already seen that yes, amino acyl synthetases they are important because they find out the correct amino acid and then they add up that correct amino acid to the correct tRNA. So that is a big challenge, right? To find out the correct amino acid from the pool of the amino acid in the cell and then to add up it to the right tRNA. Okay, so this is about that only. Now here this question talks about the misacylation that could be done by the amino acyl synthetase for threonine, right? While adding the amino acid to its tRNA. So every amino acyl synthetase that will be adding up the right amino acid to the right tRNA. However, it has been asked that misacylation may occur. It may add up some wrong amino acid to the respective tRNA. So what are the statements which we could consider correct related to this thing? So here option 2 would be the right answer. Statement B and C, right? So statement B, what do we have? It possesses a distinct editing site that preferentially deacylates the misacylated tRNA, the tRNA for threonine. So yes, this is correct. Okay, let me tell you that there remain an editing site in addition to the catalytic site in amino acyl synthetase, where if any wrong amino acid gets attached. So amino acyl AMP that will get hydrolyzed, right? If you know about the charging of the tRNA process, okay, so there you might have learned about the adenylation, means the steps in which the charging occurs. So while amino acyl synthetase will be adding up the amino acid to tRNA, adenylation occurs, means AMP that is attached to the amino acid, right? So that will all happen at the catalytic site, but beside that there will remain an editing site. Okay, so if a wrong amino acid that gets attached, so that will be hydrolyzed. Okay, so this will be helping with the proofreading that there should not be a incorrect attachment of the amino acid through the amino acyl synthetase to the tRNA. Next, we have statement C that the editing of the misacylated tRNA threonine that occurs frequently in cis before the release of the misacylated tRNA threonine. So what do we mean by this, right? So here like I have already explained that amino acid TRNA synthetases to maintain the fidelity during protein synthesis by selective activation of cognate amino acid at the amino acylation site and hydrolysis of misformed amino acid TRNA at the editing site. Now next, why it is known to be occurring in cis? Because amino acid synthetase that itself is doing it. Okay, so that is known to be done in cis manner, right? Threonine tRNA synthetase, this is amino acid synthetase, it misactivates serine. If it misactivates serine, means if it adds up the wrong amino acid, not as per the nature of the amino acid synthetase, then it will be utilizing an editing site, which is C182 in E. coli to hydrolyze the serine tRNA threonine. So that means this is occurring within the amino acid synthetase. So that what has been mentioned here, that it occurs frequently in cis before the release of the misacylated tRNA threonine. If it will remain misacylated, so it will get hydrolyzed. If it will be correct, so it will be kept. Okay, and that amino acid will be further attached to its respective tRNA. So again, that this question is not that difficult, but then it was not that simple. If you know about the complete mechanism, how the amino acid that is attached through amino acid synthetase to the tRNA, if you know about the fidelity mechanisms which are followed by the amino acid synthetase, then only you could have answered this question. Okay. Now, let us move to our next question, question ID 67. Okay. So here, this question has merged the concept of mutations from Unit 8, right? Definitely, because in Unit 3 also we have this mutations and the DNA repair mechanism. Okay. So now let us find out that what this question is all about. The question asks that a nonsense mutation in the protein coding region of an upstream gene of a group of genes in an operon often leads to depletion of the downstream gene products. That is true. Okay. If any mutation will occur, so the downstream products, they'll get impacted, right? So this is a classical example of the phenomenon of polar effect. Yes, so polar mutations, they affect the expression of downstream genes or operons. So we had to choose that which combination of the statements they are true about the phenomenon. So this is very important, right, to check whether they have asked you to choose the combination for correct or incorrect statement. So here option 3 was correct. That means statement C and E are correct. Statement C says that while the presence of termination codon in the upstream ORF may deplete the ribosomes that travel down to the downstream ORF. ORF, I'm sure all of you know, ORF is open reading frame. Okay. So the ribosomes, those are traveling down to the downstream ORF. The depletion of ribosomes there will allow the loading of the rho factor. Okay, so rho factor, now see, they have again merged the concept, right? Here they are talking about the transcription termination. Rho factor, you know, rho factor that helps with the termination of the transcription. Okay, so that will result in the premature transcription termination. So what this statement is saying, that if there will be any mutation, right, presence of termination codon in the upstream ORF, right, so that will bring reduction in the number of the ribosomes that they will be traveling down to the downstream ORF. So depletion of ribosome downstream of the nonsense mutation, that will allow the rho factor to result in premature transcription termination. Okay, so yes, this is correct. Then a statement, we have statement E here, right? Presence of the suppressor tRNA reading the nonsense codon generated by the mutation diminishes the consequences of the polar effect. This is again true. Okay, so you must read and you must learn about the effect of the suppressor tRNA, because if you will compare these two statements here, D and E. So here in statement D, they have actually mentioned that this is essential for causing a polar effect of mutation. Whereas in statement E, they have mentioned that this is diminishing the consequences of polar effect. So these two are contradictory, right? So only one of them could be correct. So that is option E. So option C and E, they are correct, right? Okay, so this is where we can see the explanation, right? As I've already mentioned, that polar mutation, they'll be affecting the expression of the genes or the operons downstream, right? So that makes statement A and B as incorrect. Okay, because in statement A, you can see it has been mentioned, it does not affect the process of transcription, right? Whereas in statement B, they have mentioned that row independent termination that will be seen. Okay, so that makes statements A and B as incorrect statements. Now let us move to our next question, question ID 68. Regulation of mRNA translation is a major mechanism that maintains stoichiometric availability of ribosomal proteins. I repeat, ribosomal proteins. See, R proteins. R proteins, they combine with rRNAs to form what? To form ribosomes. Am I right? Okay. So the question is that how the level of R proteins that is being maintained. Okay. So translational regulation for these rRNA proteins is facilitated by general occurrence of R protein genes in several operons containing multiple genes. So which one of the following represents an established mechanism to ensure optimal productions of the R proteins in E. coli when the R proteins accumulate in free forms? R proteins, they are getting in excess, so how they'll be controlled? Okay, so here the answer will be statement 3. Right now, why it is so? Because statement C, we can see that free RNA protein bind to the mRNAs and down regulate their translation. Yes. Interestingly, what happens that these are proteins which are formed, right? These proteins, they actually show translational feedback mechanism. They are part of translational feedback mechanism, where what happens that each operons for these genes, it is encoding a single bifunctional R protein, right? And this bifunctional R protein, it can bind not only to a high affinity site on 16S or 23S rRNA. Now, what are 16S or 23S rRNA? They'll be making up the prokaryotic ribosomes, right? So to them, the R protein they should bind, and then the ribosomes that should be formed. So yes, these are proteins, they have an affinity towards 16S or 23S rRNA, but also a lower affinity site on their own mRNA from where they are being produced. Okay, so when they get in excess, they'll bind to the mRNA and they'll down regulate their own production. Okay, so that is why this is known as translational feedback mechanism. Okay, so by inhibiting translation of R protein operons only, when the R protein concentration exceed that of rRNA, the translational feedback mechanism couple R protein synthesis to rRNA synthesis and could be sufficient to account for the coordination of the synthesis rates of all the ribosomal components with changes in nutritional conditions. Means as per the requirement, this translational feedback mechanism could work, that could check over the concentration of the R proteins, right? So that was the last question we had discussed. So I hope you found this session helpful. I hope you got an idea that what all topics you should cover if you are planning to prepare for the next CSIR exam paper. Okay, do let us know in the comment section if you have any confusion over solving any of these questions we have discussed in this section. We'll be glad to help. Thank you. Take care.